I'm going to be talking, my name's Daniel Howrigan. I'll be talking about meta-analysis, and fortunately a lot of the practical portion of the course is through, so I'm going to kind of talk through things. I will show some reference to how you would conduct a meta-analysis in Plink, but most of this, kind of sit back, you can kind of relax, stop the speed typing, and kind of get an idea of what you're going to do. Say you've done a GWAS. Everyone, we found the SNP. We did it. We can go home, publish in Nature. You know, did you find anything? Did you find everything? Maybe you're using some old data. You haven't scanned everything. Do you even believe what you have found? Right? And by and large, a lot of the work that's been done in the past, kind of the decades coming from candidate genes to GWAS to these large analysis to what Ben was mentioning. Now we've got 40,000 cases and controls for schizophrenia. The picture moves around a lot. Things pop up, people think something's significant. You look at the bigger data, it's not significant. And a lot of the work that's been kind of pushed in more recent times is all these consortiums and collaborations, and this is where meta-analysis becomes a really important tool to accurately say, okay, you've done your GWAS, we've done our GWAS, and now we want to put it together, see if we can maybe find more things, make sure the things that we found are real or not. And so what I'll be talking about is why we run a meta-analysis, some of the preps and issues, some of the fundamental statistics that go into combining these p-values that you generate from your GWAS and other GWASs, and then a quick foray into the software. Plink has a meta-analysis package, so don't worry, you don't need to learn anything too new. The main question, why? Well, as Chris was talking about power is kind of the central theme in a lot of the genetics literature. What we found, particularly for many of these complex diseases that effect sizes are small, we need these large samples. What's a great way to get a lot of this together? Meta-analyze a lot of the statistics, and also effect size estimates. We're not just concerned with whether it's significant or not, whether it's real or not, but how real, how much of an effect does it have? Is it an ApoE that explains a large proportion of the variance, or is it something much smaller? Say some of the newer regions that we're finding in these really large things, they have very small effect sizes, despite the fact that now that we have these giant samples, we can detect those effects. And what we're going to do, we'll get into these a little bit more as we go along, but what you're really using is the summary statistics in most cases. What Chris was talking about, say with these tests, when we think of meta-analyzing data, when we have all the raw data together and you run kind of a stratified analysis controlling for different data sets or whatnot, a lot of times you don't have access to that, but you will have access to the summary statistics. And this is an important thing when you're dealing with IRBs and who's allowed to do what. A lot of times the raw data is not allowed to be moved around, but we can give you our file of all our summary statistics from our GWAS. And so a lot of people, Wernery is a good example. He's done a really large meta-analysis, and he's collected from all sorts of different groups. And for him, he doesn't need to grab all the raw data. He can grab a lot of summary statistics files and get pretty much a precise answer. There are a few subtle things like population stratification and issues that you might be worried about when just collecting summary statistics, but by and large, you will arrive at the same answer whether the data is combined from the raw data or the summary statistics. So kind of going to the steps, the obvious one, identify the relevant studies. Make sure that when you're putting these things together that people have measured everything kind of similarly. You want to make sure that the population's similar. A lot of these artifacts that we're worried about will pop up when you get things, and you got to control for that. So there's a somewhat different type of QC. One of the things, say the uniformity in your phenotype, which I'll talk about, and different file formats. As many of you know, once you start collaborating with a bunch of different people, you realize just how quickly everyone's doing things a little bit differently, making sure that everyone's on the same page and stuff. And, you know, from a logistics standpoint, you know, these things can be a little harder than you think at the face of it. For example, phenotype, something obvious, like height. You know, if you collected height and one study was looking in the metric system, the other one's using inches, you know, if you overlook that, all of a sudden you're finding all sorts of stuff, but it's just because your phenotypes are off in some fundamental way. One of the ways that you can kind of control for this, maybe with things that's not so easy as centimeters and this, is that you make sure that if you're getting a lot of studies together, make sure that your collaborators what you would do is like normalize your phenotype. So instead of thinking about the raw height, you just say, oh, let's normalize everything within data set. That's another way to get around this. But you know, in the case of gathering a bunch of different studies, you want to make sure that everything is fitting in the right place and that you're not interpreting things as just a measurement error artifact. Clearly, sharing summaries, this probably takes longer than any other part, only because you're waiting on the laziest collaborator to send the files over, right? So I'm sure we've all been there in some way, shape, or form. But kind of back with the QC stuff, this is something this is a bit of a segue out of this, but one of the important things when you think about one of the big issues that pops up, and kind of getting back to the fundamental aspect of looking at these markers and kind of the variation in DNA, is that a lot of times the way these chips were designed and kind of the progress through these studies is that a lot of the biology or the biochemistry was kind of jumping around from the forward strand and the reverse strand, and then what ends up happening downstream is that now that everyone's trying to put all their data together, people ran into this problem that, oh, I used an Illumina chip and they're all on this strand. Oh, I used an Affy chip and, well, some of them are on this strand, some of them are on that strand. And what ends up happening is that, you know, sometimes that marker could be an AC marker, but in a different chip, it could be a TG marker. And this is a very real thing. Now, normally it'd be easy. It's like, oh, well, clearly that same base position, you know, on this chip is TG, and you tell people, okay, make sure everything's on the forward strand. But you get SNPs like this, AT, and if you flip them around, they're TA. And you don't know whether this population, sometimes if the A's here show up only 5% of the time and this one's 95% of the time, you say, oh, look, your T's showing up 95% of the time. We're fine, just flip them over. But a lot of times you don't quite know what's going on here. And one of the things that, like, you can use this allele frequency, you can use things like linkage disequilibrium, which we didn't talk too much about here, but one of these issues is ambiguous SNPs, and this is a little bit of an aside, but it's something that I think all of us who have done a lot of this this work have ran into this issue, and it's not easy to resolve. And so just to kind of, when you're thinking of meta-analysis, this is a big issue. There isn't an easy solution, unfortunately. A lot of times people just throw these SNPs away. But anyways, back to meta-analysis. What we do once you have your summary statistics, you combine them. What you're also going to look for, which I'll get to this in detail here in a minute, is to look for heterogeneity, to see that maybe there's a lot, or are we seeing a consistent pattern in our data, and there's ways to test for that using statistics. And then finally, publication bias. So if you're looking into the literature at, say, a particular candidate SNP, one thing you have to worry about is something like publication bias, where there's a bias in the fact that, you know, it's much more likely to publish something that you saying something's happening, then nothing's happening. So, all right. So I bet that's how a few of you people feel at this point about getting into the equations at this hour. But this is one of the conceptual statistic for combining things together. You got this, where this equals this and that equals that, blah, 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 all this stuff. I'll get into the details. I just like to scare you like most stat types do by spitting out an equation and then defining it down the ways. And this z is kind of from a conceptual point. If you think about your test statistics that you get out and their resulting p-values, is that, you know, by and large, most of them, nothing's happening. And what happens when that happens, you get this nice null distribution. And Ben very eloquently talked about this nice null distribution and how things fall out. Well, along that thing, we like to think of our statistics as following this pattern, and a normalized pattern along this normal distribution is called a z-score. You may have heard of this in your intro to stats class. You know, z-scores of 1 and 2 are this many standard errors away from the mean, and everything's nice. And this also provides a really good way to take all your statistics and normalize them along this pattern, and take p-values from different size studies and take them all together and make sure, you know, that we've standardized them correctly. So what I'm going to do is go through an example here. We'll take a SNP. Let's say the one we found in your GWAS was this one, and you've got kind of similar output. Here's your minor allele frequency that you got from the .FRQ file. You've got a lot of the stuff that comes out in the .ASOC file, and you've got your statistic, your p-value. Oh, look at this one, might be genome-wide significant. You've got a nice odds ratio, as you can see. You know, in the smaller sample sizes, everything looked nice, and then the BroadE genotyped 15,000 people, and we're not sure. Okay, it might be in the same direction. It's not nearly as significant as before. What's going on? Let's meta-analyze it, right? And what we're going to do, that first thing, we want to get these z-scores. What we do is take those chi-squared values that you got from your association statistic. What you do is you take the square root of that. And so now it's normally distributed. So we convert these into z-scores. But you want to make sure that the direction of your test is in the same direction. So we're assuming, okay, we're looking at the minor allele. It's all the same, say t. And one nice thing that we look at the odds ratio, they're all above 1. That means it's all risk. This 5 to 8% allele, depending on your sample, it's all conferring risk. And so we can say, okay, all these z-scores will be positive. If these were below 1, then this would be a resulting negative z-score. So we got our z-scores. Now we want to convert our sample sizes into weight, right? We want to make sure that this sample, which has 15,000 people, is going to be more important than this sample that just has 1,000 individuals, right? And so we just take the square root of n. This converts this into a weight, and then now we can kind of go to that final statistic. There are different ways of doing weights. I brought this up. If there's unequal case control balance, right now we won't go into that, but say this 5,000, 10,000 thing, if it was maybe 7,500, 7,500, you might do a different, there are some different equations that can make adjustments and for that stuff. But for the moment and for most purposes, it doesn't make too big of a difference, but we're just gonna normalize from the entire sample size. So hopefully I'm not running too fast here, but we're gonna calculate our meta-weighted z statistics. So we got our z-score and our weight. Let's do our numerator, multiply them, add them up. We do our denominator, square each of these weights, add them up, and then take the square root. We've got our weighted z, and then what we can do, if anyone still has R open in their thing, is if you could do 2 times p norm, and this is the value that we get for our weighted z. If someone could tell me, so I'm still making people do stuff, somewhat. You guys are probably too tired, but what this is doing is what's nice is that R is not just a calculator. It also has these nice statistical distributions built in, such as the binomial and the chi-square and the normal distribution. And what you can do is you can take a z statistic and just say, where does this fit along the normal distribution? Does anyone have this answer here? 4 times 10 to the minus 6. So you take that. Unfortunately, you put them all together, it's not genome-wide significant. As we thought, maybe the finished sample, things were looking good, but it might not be real. So this is, hopefully everyone kind of has an understanding here what goes on. There are different ways of doing this. So this example is a case control type of analysis. You can do things like logistic regression. You can kind of get all those different parameters together in terms of controlling for different principal components, and this is kind of a downstream thing. Yes.
What is the threshold for genome-wide significance?
As for genome-wide significance, 5 times 10 to the negative 8th, and so this has been established over the years, and as the studies have gotten bigger and bigger, and actually it's been quite robust. Where does it come from? So it's the multiple testing burden that you face when looking across the genome. So if you're doing the genome-wide analysis, is kind of a sense of an agnostic analysis of, we're just going to look at everything, and we're not going to assume any particular biology and only looking at this and that. And when you're doing that types of tests, and just like paying the penalties of doing multiple tests is in degrees of freedom, when you do a lot of multiple testing, you're going to find that stuff is going to pop up. And what they found by doing a lot of work in terms of looking at the distribution of test statistics as we've gotten more and more into the genome, and as we've mapped finer and finer things, that it starts to reach a limit at about 5 times 10 to the negative 8, where these are the number of independent signals that we're seeing in the genome. And it's not to say that everything below it is not real and everything above it is. Like any statistics, there's noise around, and a lot of times, what we've seen as a really great example in the emerging really large studies is that when you have these smaller studies, much of those signals could be real, they just do not have the power to be detected. And so I won't get into detail, but this can be used for quantitative traits as well, or you can actually pull betas out from a logistic regression and look using the variance of the term and not just the sample size to create your weight. This would be what comes out of a .QSOC, and actually it gives you a beta, this guy right here, and it gives you a standard error which Plink actually can plug in and create these weights for you, and it's nice there. And very similar type of deal. I won't go into the example about this, but not just Z's, but betas can be used. And then there's another class of meta-analysis statistics, more traditional ones, called the Fisher's test and just summing of chi-squared values. And these are directionless methods. These are kind of asking, we don't care what direction it's in. We just want to see if something is happening. And there's a little bit more freedom, and to say that, well, you can look at different things, where these weighted statistics were much more kind of confined to what's this marker position doing. This can look at different things, but what you'll see, these methods by and large are fairly similar. If I took those p-values that we saw in that table and did the math, ran it through, and calculated a p-value, you would get something like, I think, 1 or 2 to the e to the negative 10. So it's going to be much more significant if you do this Fisher's combined p. And one of the things that changes here is this is a 6 degree of freedom test because you've done 3 comparisons and each one uses actually 2 degrees of freedom. But what we've seen, let me see if I can, I should have actually just put up the p-values to save you time, but we see a more significant p-value using this method. And the reason we see it is because we're treating each of these p-values as if they were equivalent. And what we know from looking at the weighted z is that, wait a second, this one is much more important than this guy because this one is from 15,000 people and this one was only from 2,000 people, right? And what this kind of highlights is that this method, while being robust in the sense of this is an accurate way to combine p-values, is it's kind of implicitly saying that we're going to give importance to each p-value equally. And if I would take, say, this first study that we got this p-value here and kind of compared, let's just take this and say let's combine it twice. And then what we want to also compare is this weighted z. So I'm going to do a comparison between this p-value, weighted z. I did all the math for you. And when you actually take equivalently sized studies, what it turns out is that this gives you a lower p-value than this one. And so by and large, this is actually a much better powered test. And it's more accurate. So for the sake of jumping around different p-values, this is a more accurate test. This takes in more information, and if you had equal size samples, this would actually produce a lower p-value. But in this previous case, this p-value is highly inflated because you're treating these equally. So details, whatnot. If I do the sum of the chi-squares, we actually get very similar, this should say chi-squared, but we actually get very similar p-values to the Fisher's combined, but for the sake of giving you guys an idea of the different meta-analytical tests out there. Here's a visual representation. I think this is these forest plots are a really great way to kind of visualize and see what is going on. That in each individual study, you'll see those confidence intervals. Chris kind of talked about the 95% confidence interval. This is a function of the error around your estimate there. So your odds ratio is, jumping around from study to study. Then when you combine them all and you do a meta-analysis, you get a nice thing, and you can see that these error bars have shrunk quite dramatically because you've combined all these tests. And now you can see what was kind of a really small effect size, converge now that you have the power to say, look at the consistency across all these studies. That yes, it's small, but as you've combined them all, done a meta-analysis, you can arrive at this significant p-value. Here's another example of a forest plot provided by Bernery and some of the data he was looking at. And just to be a stickler, I like this. This is a good example of, hey, what's going on right here? What's this guy's deal? Everything was nice, and it looks significant at the end once you combine all these studies together. But what's with this? Should we keep this in? Is this a true outlier, or is this maybe, okay, it's out there, but it might not be? Well, one of the ways to test for that is the test for heterogeneity. And more math, more equations, somewhat similar stuff, but I think the focus is right here. And this is kind of, this was our kind of overall mean. You could think of this as that overall Z or that overall beta. And now we're saying, how does each individual one vary around that, right? And this would kind of, in a visual way, here we've got our overall. We want to test this distance across all these guys. And what you're going to aggregate is, hey, is the spread around this mean? Is this unusual? You know, maybe if we had stuff jumping all around here, we might say, you know what, this is an unreliable. There's a lot of heterogeneity in our data. And what you would often probably see is that across many, many different SNPs, you would see this. If there's an issue, it would more likely be genome-wide, that you would see a lot of heterogeneity. And fortunately, Plink, when you do these things, provides all these tests for you. Before I show you that, this is publication bias. So if you're getting these p-values and these weights and estimates just from the previous literature, as opposed to sharing summary statistics across groups, one of the things you might want to do, this would be equivalent to kind of the sample size, and then this would be the estimates, is that when these sample sizes get ramped up, you should see somewhat of a funneling fashion. But if you see that a lot of, say, earlier studies, you know, are unusually in this end, then you might be worried, hey, look, you know, there might be publication bias. Those initial reports were all favoring this particular thing. There's kind of a file drawer problem. All the people who didn't find said thing, you know, didn't publish their master's thesis, and all the ones who did got it out there. But one of the nice things with GWAS being an agnostic tool to just search across the genome, one of the nice things about all this is that we're just kind of looking everywhere. And so we're already paying such a penalty for all this multiple testing. One of the nice things that comes out of it is that when we've looked at everything, you know, we can look at everything across all these studies. And if you do imputation, where you can kind of impute all these different markers across the genome, you know, you have this consistency across studies that, you know, the bias tends to be removed because we don't have kind of a certain hypothesis driven approach. And if we went to Plink, this would be the area. You could just go on there. One of the nice things, you've got this is a bit too small, but you literally go in there, you write down your .soc files in a list, and you get something that comes out that says plink.meta, and you get your combined p-values, and you get your resulting Q statistics for heterogeneity, a combined estimate for your odds ratio, and it's pretty straightforward. One of the other programs that uses this is Metal, and I think for the sake of time, this will be it. Do you still believe it now that you've done your meta-analysis? Yeah. Sorry, I just had to throw that in there. All right, one question, then if Caitlin, if you want to start setting up. Yes.
I have a feeling there's one more shortfall on thinking about rare variations rather than common variants. So it's about 20 minutes. We'll be a few minutes over the 5 o'clock deadline. If that's okay with everyone, we'll go.
All right.
No dissent. So is there like a central repository for these summary statistics, or where do you get them?
There are for some and not for others. It kind of depends. There's dbGaP has a lot of stuff. This database for genotypes and phenotypes has a lot of raw data and stuff. A lot of times if you're looking at a particular signal that's interesting, you don't have to go too far, but for the majority of cases so far, I would think you actually have to talk to the collaborators themselves.
So even though the studies are published, the data is not accessible?
They should be accessible if they're following their protocols and guidelines of data sharing. And more it's like that. I think kind of the old guard of you know, kind of keeping to yourselves, particularly in the arena of complex trait analysis. Everyone's kind of finally coming to and saying, hey, we're not going to find anything unless we share. Mm-hmm. Mm-hmm. And I believe with all those, you can also get the raw genotype data. And you know, one of the nice things with the raw genotype data, if you say you're interested in something that's not just association, maybe you're interested in a pathway analysis or CNVs or you know, inbreeding, stuff like that. You can do things with the raw genotype data that you can't do with just the summary statistics. Mm-hmm.
So I looked at the formula only treating the new GWAS.
Maybe. I don't know. I couldn't tell you off the top of the bat. I mean, a lot of what we've used in this field is the weighted statistics. And so I believe that, yeah, can you get odds ratios from combined Fishers? No. It's very p-value oriented, and it's very structured on the distribution of p-values.